Hi, I'm Professor Johnson, and in this video we are going to be talking about the basics of meiosis. So meiosis, you may know, is the division of cells that leads to the production of sex cells. It's going to be similar in many ways to mitosis, but different in important ways that are necessary for the cell to become a sex cell. So first, going through a few terms. Gametes are what are produced through meiosis. So the gametes are your sperm cells and your egg cells. Now these are haploid, meaning that they only have half the chromosomal number. So they don't have the pairs of chromosomes that you would normally see in a diploid cell. This over here, after the cell is fertilized, is the zygote. It is diploid, so it has these pairs of chromosomes. So this is what the cell looks like when it's diploid. This is what the cell looks like when it's haploid. The zygote will give rise to many different types of cells. Some of the cells that it gives rise to are the somatic cells. So soma means body, so these are the cells of your body that aren't involved in reproduction. The other cells that it gives rise to are the germline cells. So the germline cells are the cells that will divide to eventually give you the gametes. So the gametes are produced when the germline cells go through meiosis. So to get ourselves reacquainted with the chromosomes, let's do a little bit of review on the structure of chromosomes. So each of these chromosomes are made up of the two double strands of DNA. Over here on this side we have a pair of chromosomes, so these are homologs, or homologous chromosomes, meaning that they have the same genes on them, but they might not be the identical versions of the gene. So they may have different alleles on these chromosomes, but it'll be the same genes that are present on both of these. Now, one of these chromosomes here originally came from your father, from the sperm cell, and the other one originally came from your mother, so the egg cell. Now, when these cells replicate, they start to look like structures like this. So now we will have the sister chromatids. So the sister chromatids are connected here at the centromere. So you have the one chromosome here, and you have the other chromosome here. And these are actually considered the same chromosome because they are sister chromatids and they're attached. So this is considered one chromosome. This is considered a second chromosome, but you have twice the genetic material that you have over here because the DNA has replicated. So you have what will eventually be two separate chromosomes when they are replicated. So homologs, homologs, sister chromatids, sister chromatids. Now, just a little bit of an overview on meiosis. So meiosis consists of two divisions instead of just the single division that mitosis consists of. The first division is the reductive division. It's considered the reductive division because this is the division from which the cell will go from being a haploid cell to being going from being a diploid cell to being a haploid cell. So after the first division, the cell is going to be haploid. The second division is going to further separate it. So we've got to separate the homologous chromosomes, and we also have to separate the sister chromatids. So the homologous chromosomes get separated during the first division. The sister chromatids get separated during the second division. DNA replication does occur during meiosis. It occurs before the first division, but there is no division between the two, or there is no replication between the two divisions of meiosis. Something that's unique to meiosis is the phenomena of synapses. This is where the homologous pairs of chromosomes actually align, and then crossing over, where they'll actually swap genetic information. And the other thing that's important about meiosis is the phenomenon of independent assortment. So this is looking at how the different homologs align is going to be independent of how the other homologs align. And we'll see this as we go through. So first of all, we have synapses. So synapses, for each of the homologous pairs of chromosomes, we're just showing the 2 chromosomes here for simplicity. Most organisms will have more than just these 2 chromosomes. They actually line up on the cell on top of one another. Now I'm going to be showing this a little bit different to illustrate a different point, but know that both of the sister chromatids are going to line up with, on one chromosome, are going to line up with sister chromatids on the other homologous protein. So it really looks like this, where you have this one sister chromatid on top of the red one on top of the blue sister chromatid on the other chromosome. And same here on this side. So you are going to have synapses on both sides. I don't really show that on these next slides. So when you have synapses, you're going to have these regions where you actually have a covalent bond change such that this arm of the chromosome gets swapped over onto the other chromosome. So you have this blue information coming over here, and on this red chromosome here, you have the blue information coming over and it swaps over, and you have the red information swapping over to the blue. So whereas this used to be a completely blue arm, it now has a red region to it. And this is going to happen several times on both of these sister chromatids because they're lined on top of each other, remember? So it's happening multiple times on both of them, so you're going to have a lot of different swapping going on in each of the places. I'm only showing the one spot for simplicity. And this is going to happen on all of your chromosomal pairs. So you can see that I did it on this big pair of chromosomes as well. Now, during metaphase, I want to point out that during meiosis, these aren't going to align. So that you'd have one of these homologs lining up in the middle, and the other one would line up in the middle as well, instead of having them line up together. So they're actually pairing in meiosis compared to mitosis, where they did not actually pair, and that's important. So this pairing means that it's actually going to be the homologous chromosomes that separate, unlike in mitosis where it was the sister chromatids. So you actually pull those apart. Notice that when you pull that apart, this genetic information, so where these were crossed were known as chiasmata. These chiasmata then break, and as you break these chiasmata, some of the genetic information that was originally on this chromosome over here gets put on this chromosome. So this is now more unique than what it was, and this sister chromatid will no longer be identical to this sister chromatid because you now have this region right here that is different over here. Before, because these were due to DNA replication, these were identical. Now these aren't different. So this is the first thing that contributes to the genetic diversity of your offspring. So all these gametes are going to be different now because you had crossing over occur. Now, the second thing that contributes to this is the independent assortment. So I'm showing two different possibilities here. One possibility are that the blue chromosomes, which I'll be calling the paternal chromosomes, line up on one side of the cell, and the maternal ones, the red ones, line up on the other. So now when these separate, you will have the red chromosomes on one side and the blue chromosomes on the other. Another possibility for how they could align is that the paternal chromosome and the maternal chromosome of a different homologous pair could line up on one side, and then you'd have the opposite on the other, and so this would produce different cells as well. So you can see how this leads to 4 different gametes that are going to be produced. In reality, there's going to be a lot more different possibilities because I'm only showing the simple situation here where you have 2 chromosomes, but the actual number of possibilities for independent assortment is going to be 2 to the n, where n is the number of chromosome pairs. So in this case, it's going to be 2 to the 2, so you can see that you end up with 4 different possibilities here. If in humans, it would be 2 to the 23, since you have 23 different pairs. So it leads to a lot more diversity in the different kinds of pairing that can actually go on. And so this is actually what the chromosomes will look like, or what the cells will look like after meiosis 1. You're going to have them separated into different cells. Notice that the homologous pairs are no longer in the same cell. but the sister chromatids are still attached. So in the second phase of meiosis, you're actually going to have to separate the sister chromatids. So this is going to look a lot more like mitosis, where you have them aligning in the middle of the cell, and then these are going to pull to one side, these are going to pull to the other side, and you're going to have these different chromosomes or different cells being produced from this. And remember, you have two cells here since you went through one round of cell division that produced two different cells. So now you have the two different cells that are possible. And so this is what it looks like after meiosis 2, where you're going to have these separated onto each side. Now I want to point out that because crossing over has occurred, it won't be equivalent with this. So in this slide, I'm showing the four different cells that are being produced from these. In this next slide after this, I'm just looking at these and seeing how they could be oriented differently. So you can see this is in the middle here is how they're originally oriented in the previous slide, where you have the two chromosome sister chromatids that had the crossing over occur on one side, on the same side. You can have them aligned so that that isn't the case. And in reality, crossing over is going to occur multiple times. So you're going to have crossing over on both of these, but how one homologous pair of one chromosome aligns up in the middle of the cell is not going to affect how another chromosome lines up in the middle of the cell. So this also contributes to the genetic diversity of the offspring. So what's really contributing during meiosis is the independent assortment of chromosomes of the homologous pairs of chromosomes during meiosis 1. The fact that you're also having something similar going on during meiosis 2, the chromosomes are now different because of crossing over, or the sister chromatids are now different because of crossing over. Before they were identical, after crossing over they're different. And crossing over is the other thing that contributes to the genetic diversity. Alright, I hope this was informative to you, and I'll see you guys next time.